I see the light. J.O.L.A.L.F is poison, such a death. I lost my head, shit, who am I think I'm dead. Falling through the void, flying through the static. We can share a casket, my synapse is neurotic. My synapse is neurotic. My synapse is neurotic, yeah, my synapse is neurotic. My synapse is neurotic. She's about to turn up in the club, and after that she'll come and turn up in my tub. It's kinda like we're turning on the microwave. Oh, she to pray, all I know is what she say. I love the way you moan, just keep it low, cause the neighbors might know. They might be listening to my tones. I spit the heat, they call me an inferno. J.O.L.A.L.F is poison, such a death. I lost my head, shit, who am I think I'm dead. Falling through the void, flying through the static. We can share a casket, my synapse is neurotic. My synapse is neurotic. My synapse is neurotic, yeah, my synapse is neurotic. My synapse is neurotic.
An introductory neuron course for the J.O.L.A.L.F would explore the fundamental unit of the nervous system, the neuron. It would cover the basic structure including the cell body, dendrites receiving signals, and the axon transmitting signals. The course would explain how neurons communicate through electrical signals called action potentials and chemical signals via neurotransmitters at synapses. Key concepts like resting potential, depolarization, and the role of ion channels would be introduced. Furthermore, the course would likely touch upon different types of neurons: sensory, motor, interneurons, and the basic organization of neural circuits. A neuron is the fundamental signaling unit of the nervous system, responsible for receiving, processing, and transmitting information. It consists of a cell body soma containing the nucleus and organelles, branching extensions called dendrites that receive signals from other neurons, and a long projection called an axon that transmits signals away to other neurons or target cells. Communication between neurons occurs at specialized junctions called synapses, where chemical messengers called neurotransmitters are released from the presynaptic neuron and bind to receptors on the postsynaptic neuron, initiating a new electrical or chemical signal. Electrical signals within a neuron, known as action potentials, are rapid changes in membrane potential that travel down the axon. Neurons exhibit a resting membrane potential maintained by ion pumps and selective permeability to ions. When stimulated, the membrane potential can change, leading to depolarization and the generation of an action potential if the threshold is reached. Different types of neurons exist, specialized for various functions such as sensory input, motor control, and interneuronal communication within the nervous system. Statistics: the adult human brain contains approximately 86 billion neurons forming about 100 trillion synaptic connections. These neurons, diverse in type and morphology, sensory, motor, interneurons, communicate through electrical action potentials and chemical neurotransmitters at synapses. They are supported by roughly an equal number of glial cells. While varying in size and shape, neurons can transmit signals rapidly. Synaptic connections exhibit plasticity, changing with experience, and neurogenesis occurs in limited adult brain regions. Despite being a small fraction of body weight, the brain consumes a significant portion of the body's energy. An artificial neuron, the fundamental unit of artificial neural networks, is a mathematical function inspired by biological neurons. It receives one or more inputs, each associated with a weight representing its importance. These weighted inputs are summed, and a bias term is often added. This sum is then passed through a nonlinear activation function, which determines the neuron's output. This output can then serve as input to other artificial neurons in the network. The activation function introduces nonlinearity, enabling the network to learn complex patterns. The weights and biases are the parameters that are adjusted during the learning process to optimize the network's performance on a given task. Statistics: the number of artificial neurons in a neural network can range from just a few in very simple networks to billions or even trillions in large-scale deep learning models like large language models. Similarly, the number of connections, or weights, between these neurons can also vary dramatically, often being orders of magnitude larger than the number of neurons themselves. For instance, some state-of-the-art large language models have hundreds of billions or even trillions of parameters weights. The arrangement of these artificial neurons into layers and the way these layers are connected define the network's architecture, which significantly impacts its complexity and capabilities. Deep neural networks, for example, have multiple hidden layers, allowing them to learn intricate patterns. The complexity of a neural network is also influenced by the type of activation functions used in its artificial neurons, as these functions introduce nonlinearity crucial for learning complex relationships in data. Furthermore, the number of inputs each artificial neuron receives and the way these inputs are weighted contribute to the overall complexity and the network's ability to model intricate functions. The density of connections, how many neurons are connected to each other, is another statistical aspect, with some architectures like fully connected layers having high density and others like convolutional layers having more sparse connections. The resources required to train and run these networks, such as computational power and memory, often scale with the number of neurons and connections, highlighting the statistical impact of network size on practical implementation. For biological neurons, we can't directly apply Big O notation to describe their individual performance in the algorithmic sense. Instead, we consider how the complexity of neural circuits and the time for information processing might scale with the number of neurons akin to n. For instance, a larger brain with more neurons might theoretically be capable of more complex computations, but the speed of signal propagation is limited by biological factors, and the processing is highly parallel and interconnected, unlike sequential algorithmic steps. For non-organic artificial neurons within artificial neural networks, the concept is closer but still not a direct fit. While we can analyze the computational complexity of algorithms used to train and run these networks which would use Big O notation, applying it directly to the performance scaling of the network itself with the number of artificial neurons n is less common. Instead, we typically evaluate the network's performance based on metrics like accuracy on a given task, the time it takes to process an input, inference time, and the resource's computational power, memory required, which often correlate with the network size and architecture. The training process itself, which adjusts the connections between these artificial neurons, has its own algorithmic complexity that can be analyzed using Big O notation, but this is distinct from the performance scaling of the trained network. Case Study 1: Scaling of Visual Processing in Biological Brains. Concept: studies on the visual cortex across different species with varying brain sizes and neuron counts can provide insights into how the complexity and processing power of a biological neural circuit scale with the number of neurons n. Example: research comparing the visual processing capabilities, e.g. resolution, speed of object recognition in animals with smaller brains like rodents versus those with larger brains like primates might reveal scaling trends. While not expressed in Big O notation, such studies examine how increased neuron numbers and circuit complexity relate to functional capabilities. Limitations: biological systems have significant parallel processing and interconnectedness, making direct comparisons to sequential algorithms analyzed by Big O complex. Factors like neuron type diversity, synaptic plasticity, and the overall brain architecture also play crucial roles. Case Study 2: Performance Scaling in Artificial Neural Networks for Image Recognition. Concept: examining the performance of ANS e.g. in image classification tasks like ImageNet as the number of artificial neurons and layers related to N increases. Example: the evolution of convolutional neural networks CNNs from AlexNet to ResNet to more recent architectures demonstrates a trend of increasing network size, more layers and neurons, leading to higher accuracy on benchmark data sets. Studies often report the relationship between the number of parameters, a proxy for N, and the achieved accuracy. Metrics: performance is typically measured by top 1 and top 5 accuracy on the image classification task. Inference time to process a single image and computational cost flops are also often reported in relation to network size. Limitations: this scaling isn't strictly big O in the algorithmic sense, but rather an empirical observation of how performance metrics change with network size. The efficiency of training, which is analyzed with big O, and the architecture's design heavily influence these scaling trends. Neural scaling laws are an area of active research that tries to formalize these empirical relationships between model size, dataset size, and performance. Cellular case study: scaling of resource allocation in autonomous floating cells, implications for network complexity. Concept: this case study explores how resource allocation might scale with increasing complexity in autonomous floating cells, drawing inspiration from the computational demands observed in training large artificial neural networks ANS. We will consider how the computational capacity and learning potential of a single floating cell might relate to its internal resource investment, and how these demands could scale in multicellular systems forming complex networks. Hypothetical floating cell system: imagine a population of single-celled organisms capable of autonomous movement, sensing their environment, processing information through internal biochemical networks, and responding with adaptive behaviors. These cells might form temporary or permanent aggregations, creating rudimentary networks for coordinated action. Syllabus: course goal: to gain a deep understanding of the cellular processes that enable biological neurons to receive, process, and transmit information, and to explore the conceptual links to artificial neurons. Learning objectives: upon completion of this course, the learner should be able to detail the structural components of a biological neuron at the cellular level and their respective roles in neuronal function. Explain the electrochemical basis of the resting membrane potential and the key ions and mechanisms involved. Describe the process of depolarization and repolarization leading to the generation of an action potential, including the roles of voltage-gated ion channels. Elucidate the mechanisms of synaptic transmission, including neurotransmitter release, receptor binding, and postsynaptic potentials. Differentiate between various types of biological neurons based on their structure and function, with a focus on how their cellular properties contribute to their specific roles. Understand the fundamental principles of artificial neurons as simplified mathematical models inspired by the input-processing-output nature of biological neurons. Discuss the limitations of applying algorithmic complexity analysis Big O to the dynamic and parallel cellular activity of biological neurons. Relate the concepts of scaling in neural systems to the increasing complexity of cellular interactions and network organization. Course modules: concise course modules. Module 1, Neuron Structure and Membrane Potential, covers neuron anatomy, soma, dendrites, axon, organelles, and the basis of resting membrane potential. Module 2, Electrochemical Signaling, explores ion distribution, membrane permeability, the Na+K+ pump, and factors influencing membrane potential. Module 3, Action Potential, details depolarization, voltage-gated channels Na+, K+ kinetics, AP phases, propagation myelin, nodes, and refractory periods. Module 4, Synaptic Transmission, focuses on synapse structure, neurotransmitter release Ca2+, vesicles, receptors ionotropic, metabotropic, postsynaptic potentials IPSBs, summation and inactivation. Module 5, Neuron Diversity and Circuits, examines neuron classification, morphology, function, sensory, motor, interneurons and basic neural circuit principles. Module 6, Artificial Neuron Analogy, introduces the artificial neuron model and its simplified relation to biological neuron function and limitations. Module 7, Scaling and Biological Complexity, discusses why algorithmic complexity doesn't fit biological neurons and how circuit complexity arises from cellular interactions and plasticity. There is never a time zero when you decide to do something, because every neuron in the brain is driven by other neurons. There seems to be no part of the system that acts independently rather than reacts dependably. David Eagleman. The origin of the neuron is a deep and fascinating question in evolutionary biology, with evidence suggesting that the fundamental building blocks for these specialized signaling cells arose in the Precambrian era, possibly around 800 million years ago. Molecular studies point towards peptidergic secretory cells, similar to those found in modern-day Plecozoan simple, multicellular marine creatures without a defined body plan or organs, as potential evolutionary precursors to neurons. These early cells likely communicated using neuropeptides, small protein-like molecules that could influence the behavior of other cells. Over evolutionary time, these ancestral secretory cells are hypothesized to have gained new genetic modules that enabled them to develop the key characteristics of a neuron, the ability to generate rapid electrical signals through ion channels, to form elongated projections like axons and dendrites for directed signaling, and to create complete synapses for efficient communication with other cells. The first true neurons, capable of these sophisticated electrical and chemical signaling mechanisms, are thought to have originated in the common ancestor of cnidarians, jellyfish, corals, sea anemones, and bilaterians, all other animals with bilateral symmetry, around 650 million years ago. These early nervous systems were likely simple nerve nets, diffuse networks of interconnected neurons spread throughout the organism, as seen in modern cnidarians. This arrangement allowed for basic sensory responses and coordinated movements. However, the evolutionary story is not entirely linear. Comb jellies, ctenophores, another early branching lineage of animals, also possess neuronal-like cells and nerve nets, but these cells exhibit some significant structural and genetic differences from the neurons found in cnidarians and bilaterians, raising questions about whether neurons arose once or multiple times independently in early animal evolution. The absence of true neurons in sponges, porifera and plecozoans further complicates the picture, suggesting that these lineages diverged before the full suite of neuronal characteristics evolved. The development of the nervous system in bilaterians marked a significant evolutionary innovation. Their worm-like ancestors, appearing in the Cryogenian period around 700 to 650 million years ago, are thought to have developed a more organized nervous system featuring a nerve cord and a concentration of neurons at the anterior end, a precursor to the brain. This centralization allowed for more complex integration of sensory information and more coordinated motor control, paving the way for the vast diversity of animal forms and behaviors we see today. While the precise steps and genetic mechanisms underlying the origin and early diversification of neurons are still being actively researched, the evidence increasingly points to a gradual process of adaptation and innovation from simpler secretory cells to the highly specialized and interconnected cells that form the basis of nervous systems in most multicellular animals. Case study: the widespread similarity in the fundamental molecular components that enable neuronal signaling across diverse animals, from ion channels and synaptic proteins to signaling pathways and developmental genes, strongly suggests that these crucial elements were inherited from a single common ancestral neuron. This deep conservation of the core machinery points to a single evolutionary origin of neurons, with subsequent diversification of nervous systems arising from modifications of this shared molecular toolkit. The central nervous system (CNS) is the command center of the nervous system in vertebrates and most invertebrates, orchestrating and integrating information to coordinate bodily functions and activities. In vertebrates, the CNS is comprised of two main components, the brain and the spinal cord. Both of these vital structures are encased in protective bony coverings, the skull for the brain and the vertebral column for the spinal cord, and are further cushioned by layers of membranes called meninges and cerebrospinal fluid, a clear, watery fluid that also provides nutrients and removes waste products. The brain, the most complex organ in the body, is responsible for higher-level functions such as thought, memory, emotion, language, sensory processing, and the initiation of voluntary movements. It is a highly organized structure with distinct regions, each specialized for particular functions, although there is significant interconnectedness and integration between these areas. The cerebrum, the largest part of the brain, is divided into two hemispheres, each further subdivided into lobes: frontal, parietal, temporal, and occipital, responsible for diverse cognitive and sensory functions. Beneath the cerebrum lies the diencephalon, containing the thalamus, a relay station for sensory information, and the hypothalamus, involved in regulating homeostasis, emotions, and endocrine functions. The brainstem, connecting the cerebrum and diencephalon to the spinal cord, comprises the midbrain, pons, and medulla oblongata, which control vital autonomic functions like breathing, heart rate, and blood pressure, as well as relaying sensory and motor information. Posterior to the brainstem and inferior to the cerebrum is the cerebellum, crucial for coordinating voluntary movements, posture, balance, and motor learning. The spinal cord is a long, cylindrical structure extending from the medulla oblongata in the brainstem down through the vertebral canal. It serves as the primary pathway for communication between the brain and the peripheral nervous system, transmitting sensory information from the body to the brain and motor commands from the brain to the muscles and glands. The spinal cord also mediates reflexes, rapid, involuntary responses to stimuli that can occur without conscious involvement of the brain. Internally, the spinal cord has a characteristic butterfly-shaped gray matter core, composed mainly of neuronal cell bodies and unmyelinated axons, surrounded by white matter, which consists primarily of myelinated axons organized into ascending sensory and descending motor tracts. Case study: the central nervous system (CNS), comprising the brain and spinal cord, is the body's control center. Protected by bone and fluid, the brain handles complex functions like thought and movement, while the spinal cord acts as the main communication link between the brain and the rest of the body, also mediating reflexes. Damage to the CNS can severely impair sensory, motor, and cognitive functions, highlighting its crucial role in overall bodily control and coordination. Neurons do not directly form glands, but they exert significant control over their function. Motor neurons, a type of neuron, can directly innervate glands, meaning their axons extend to and form synapses with the cells of the gland. When these motor neurons fire action potentials, they release neurotransmitters that bind to receptors on the gland cells. This binding can stimulate or inhibit the secretion of hormones or other substances produced by the gland, depending on the specific neurotransmitter and receptors involved. For example, the autonomic nervous system, a division of the motor nervous system, regulates the activity of many glands, controlling processes like salivation, sweating, and the release of digestive enzymes through the action of its neurons on glandular tissue. Therefore, while neurons are distinct from glandular cells, They are essential for the nervous system's ability to regulate and coordinate glandular activity throughout the body. Case study: seeing and smelling food triggers brain activity that activates parasympathetic neurons. These neurons release acetylcholine onto salivary glands, stimulating them to produce saliva for digestion. This shows how the nervous system directly controls glands via neuronal signaling. The soma, or neuron cell body, serves as its metabolic and genetic control hub. Within it resides the nucleus, containing the neuron's genetic blueprint DNA, alongside essential organelles like the endoplasmic reticulum for building proteins and fats, the Golgi apparatus for refining and packaging these molecules, and mitochondria, the powerhouses generating ATP for cellular energy. Beyond these maintenance functions, the soma also acts as an integration center, receiving and processing the multitude of synaptic signals arriving from other neurons, ultimately playing a vital role in the neuron's survival and overall operational capacity. Case study: the soma, or cell body, is the neuron's control center, housing its DNA and metabolic machinery. It produces energy and proteins essential for the neuron's function and integrates signals from other neurons. Damage to the soma disrupts these vital processes, leading to neuronal dysfunction and death, as seen in diseases like ALS and viral encephalitis. Dendrites are branching, tree-like extensions of the neuron that primarily serve as the receptive surfaces for signals from other neurons. Their extensive branching pattern increases the surface area available for receiving synaptic inputs. Dendritic membranes are rich in receptors that bind to neurotransmitters released by presynaptic neurons, initiating electrical or chemical signals within the postsynaptic neuron. The integration of these incoming signals in the dendrites and soma determines whether the neuron will generate its own outgoing signal. Case study: dendrites are the neuron's branching receivers, covered in receptors that bind signals from other neurons. Their extensive structure maximizes the surface for these connections. Dendrites integrate these incoming signals, which, along with processing in the cell body, determines if the neuron will send its own signal. Axon: the axon is a long, slender projection of a neuron that typically transmits electrical signals, known as action potentials, away from the soma towards other neurons or target cells, muscles or glands. A neuron usually has only one axon, although it may branch at its terminal end to innervate multiple targets. The axon originates from a specialized region of the soma called the axon hillock, where the decision to generate an action potential is typically made. Case study: imagine a single neuron as a messenger with a vital piece of information. Its axon isn't just a wire, it's a branching network of communication lines, like a single broadcaster reaching multiple listeners simultaneously. This branching is a powerful strategy, allowing one neuron to amplify its influence, rapidly relaying crucial signals to numerous downstream partners, whether sparking movement in multiple muscle fibers for a coordinated action, alerting several neighboring neurons in a neural circuit, or signaling to various parts of a gland to trigger a widespread response. This ability to broadcast from a single source underscores the efficiency and interconnectedness of the nervous system, where a single signal can have far-reaching and multifaceted effects thanks to the strategic branching of the axon. Myelin sheath: the myelin sheath is an insulating layer that surrounds the axons of many neurons in the vertebrate nervous system. It is formed by specialized glial cells, Schwann cells in the peripheral nervous system and oligodendrocytes in the central nervous system. Myelin is composed primarily of lipids and proteins and dramatically increases the speed of action potential propagation along the axon by preventing ion leakage and allowing the action potential to jump between gaps in the myelin sheath called nodes of Ranvier. Case study: myelin is a fatty insulation around many vertebrate axons, formed by glial cells. It dramatically speeds up electrical signal transmission by preventing ion leakage and making the signal jump between gaps called nodes of Ranvier saltatory conduction. Nodes of Ranvier are periodic gaps in the myelin sheath along the length of an axon. At these nodes, the axonal membrane is exposed, and there is a high concentration of voltage-gated ion channels. Action potentials are regenerated at the nodes of Ranvier, allowing for saltatory conduction, a process where the electrical signal appears to jump from one node to the next, significantly increasing the speed of nerve impulse transmission compared to unmyelinated axons. Case study: imagine the myelinated axon not as a continuous wire, but as a high-speed rail line with periodic power stations, the nodes of Ranvier. These nodes, strategic discontinuities in the insulating myelin lamellae, are densely populated with voltage-gated sodium and potassium channels. As a depolarizing impulse, the action potential propagates down the internode, it weakens. However, upon reaching a node, the high density of ion channels allows for a rapid and robust regeneration of the action potential through a transient influx of sodium ions, followed by potassium efflux. This saltatory propagation, leaping from one excitable node to the next, drastically increases the conduction velocity compared to the continuous, slower propagation along an unmyelinated axon. These nodal regions are not merely passive gaps, they are critical amplification sites, ensuring faithful and rapid transmission of the electrophysiological signal across significant distances within the nervous system, underpinning swift reflexes and efficient neural processing. Axon terminal: the axon terminal, also known as the synaptic terminal or presynaptic terminal, is the specialized ending of an axon that communicates with other neurons or target cells at a synapse. The axon terminal contains synaptic vesicles filled with neurotransmitters. When an action potential arrives at the axon terminal, it triggers the influx of calcium ions, which in turn leads to the fusion of synaptic vesicles with the presynaptic membrane and the release of neurotransmitters into the synaptic cleft. Case study: the axon terminal, the neuron's output end, is a specialized swelling that forms the presynaptic component of a synapse. Within this terminal are numerous synaptic vesicles, tiny membrane-bound sacs loaded with chemical messengers called neurotransmitters. When an electrical signal, the action potential, reaches the axon terminal, it causes voltage-gated calcium channels in the terminal membrane to open. The influx of calcium ions then initiates a cascade of molecular events that culminates in the fusion of synaptic vesicles with the presynaptic membrane, leading to the exocytosis, the release, of their neurotransmitter contents into the narrow gap between the neurons or between the neuron and its target cell, known as the synaptic cleft. This release of neurotransmitters is the crucial step in chemical synaptic transmission, allowing the presynaptic neuron to communicate its signal to the postsynaptic cell. Cell membrane: the cell membrane, also known as the plasma membrane, is a selectively permeable barrier that encloses the neuron and separates its internal environment cytoplasm from the external environment. It is composed primarily of a phospholipid bilayer, with hydrophilic water-attracting heads facing outwards and hydrophobic water-repelling tails facing inwards, creating a barrier to water-soluble molecules and ions. Embedded within the phospholipid bilayer are various proteins, including ion channels, receptors, transporters, and enzymes, which play crucial roles in neuronal function, such as regulating the passage of ions, receiving signals from other cells, and maintaining the membrane potential. Case study: picture the neuron's plasmalemma as a vibrant, lipidic matrix, a selectively permeant boundary demarcating the cytosol from the interstitial milieu. This bimolecular leaflet, with its hydrophilic moieties oriented aquatically and lipophilic domains forming a sequestered core, establishes crucial electrochemical gradients. Interspersed within this fluid mosaic are diverse integral and peripheral membrane proteins. Ligand-gated and voltage-gated porins precisely regulate ionic flux, mediating rapid changes in transmembrane potential. Chemoreceptors exhibit high affinity for extracellular neurotransmitters, initiating signal transduction cascades. Active transporters, utilizing metabolic energy, maintain homeostasis of ion concentrations. Membrane-bound enzymes catalyze biotransformation of signaling molecules. This dynamic interface is not merely a passive partition, it's an active participant in neuronal excitability, synaptic transmission, and cellular signaling pathways, a testament to the intricate biomolecular architecture underpinning nervous system function. Phospholipid bilayer. The phospholipid bilayer is the fundamental structural component of the cell membrane. It consists of two layers of phospholipid molecules, each with a hydrophilic head and two hydrophobic fatty acid tails. In the membrane, the hydrophobic tails of the two layers face each other, forming a hydrophobic core, while the hydrophilic heads face the aqueous environments inside and outside the cell. This arrangement creates a barrier that is impermeable to most water-soluble molecules and ions, contributing to the maintenance of different ionic concentrations across the membrane, which is essential for neuronal signaling. Embedded proteins. Embedded proteins are various protein molecules that are integrated within or associated with the phospholipid bilayer of the cell membrane. They perform a wide range of functions crucial for neuronal activity, including ion channels, form pores through the membrane that allow specific ions e.g. sodium, potassium, chloride to pass through, down their electrochemical gradients, playing a key role in generating action potentials and maintaining the resting membrane potential. Receptors bind to neurotransmitters or other signaling molecules in the extracellular environment, triggering intracellular signaling pathways that can lead to changes in the neuron's electrical activity or other cellular processes. Transporters, pumps and carriers actively move ions or other molecules across the membrane against their concentration gradients, requiring energy, e.g. the sodium-potassium pump. Enzymes catalyze biochemical reactions at or near the membrane. These embedded proteins are essential for the neuron's ability to communicate, maintain its internal environment, and respond to external stimuli. Membrane receptors. Membrane receptors are specialized protein molecules embedded in the cell membrane that bind to specific signaling molecules, such as neurotransmitters, hormones, or growth factors, in the extracellular environment. This binding initiates a cascade of events within the neuron, often leading to changes in the neuron's electrical properties, e.g. opening or closing of ion channels or triggering intracellular signaling pathways that can influence gene expression or other cellular processes. The type and distribution of membrane receptors on a neuron's dendrites and soma determine its sensitivity and responsiveness to different types of synaptic input. Case study: membrane receptors on neurons bind signaling molecules, triggering internal changes like altered electrical activity or gene expression, a process that can be modified by synaptic plasticity. The type and location of these receptors dictate a neuron's sensitivity to specific inputs, and plastic changes in receptor number, function, or even type are key mechanisms of learning and adaptation within neural circuits. This dynamic regulation of receptors allows neurons to adjust their responsiveness to incoming signals over time, forming the cellular basis of memory and behavioral change. Membrane potential. Membrane potential refers to the electrical potential difference voltage that exists across the cell membrane of a neuron and other excitable cells. This potential difference arises from the unequal distribution of ions, primarily sodium, potassium, chloride, and negatively charged proteins between the intracellular fluid cytoplasm and the extracellular fluid. The inside of a neuron at rest is typically negatively charged relative to the outside, with a resting membrane potential of around -70 mV. Changes in membrane potential are fundamental to neuronal signaling. Rapid and transient changes in membrane potential, such as during the generation of action potentials and postsynaptic potentials, allow neurons to encode and transmit information. The maintenance of the resting membrane potential and the generation of electrical signals rely on the selective permeability of the membrane to different ions and the active transport of ions by pumps embedded in the membrane. Case study: high-frequency stimulation strengthens the connection between neurons. Before this stimulation, single signals cause a small electrical change EPSP in the receiving neuron, not enough for it to fire. After the strong stimulation, the same single signals now cause a much larger and longer-lasting electrical change enhanced EPSP. This stronger signal can now be big enough to make the receiving neuron fire, indicating a long-term increase in communication strength between the neurons, a key mechanism for learning and memory. In neuroscience, an excitatory postsynaptic potential, or EPSP, is an electrical change in a neuron that increases its likelihood of sending a signal. This happens when the membrane of the receiving neuron becomes more positively charged, a process called depolarization. This influx of positive charge is mainly due to sodium ions Na+ entering the neuron. As the inside of the neuron becomes more positive, it gets closer to the point where it will fire an action potential, which is the electrical signal that neurons use to communicate. Therefore, an EPSP essentially makes a neuron more excitable and prone to transmitting information. Case study: researchers stimulated a presynaptic neuron with a rapid burst of signals. Before this, single signals from this neuron caused a small electrical change EPSP in the receiving neuron, not enough to make it fire. After the burst, the same single signals now caused a much larger electrical change in the receiving neuron. This larger change was strong enough to push the receiving neuron past its firing threshold, causing it to generate its own electrical signal action potential. This long-lasting increase in the receiving neuron's response shows that the connection between the two neurons was strengthened by the initial burst of activity, a key process in learning and memory. Neurons, like all cells, maintain a specific distribution of ions across their plasma membrane, creating an electrochemical gradient. Key players in this gradient are positively charged ions like sodium Na+ and potassium K+, and negatively charged ions like chloride Cl and various organic anions. Typically, there is a higher concentration of Na+ and Cl outside the neuron compared to the inside, while the concentration of K+ is significantly higher inside. This unequal distribution of charged particles results in an electrical potential difference across the membrane, known as the resting membrane potential. This potential is typically negative inside the neuron relative to the outside, usually around -70 mV in many neurons. This resting potential is a state of disequilibrium, poised for rapid changes in response to signals. Case study: J.O.L.A.L.F and resting potential. Researchers blocked the sodium-potassium pump in a neuron. Initially, the neuron had a normal resting electrical charge -70 MV. Over time, without the pump actively moving ions, the charge inside the neuron became less negative depolarized. This shows the pump is essential for maintaining the unequal ion distribution that creates the neuron's resting electrical state. The neuronal membrane is not freely permeable to all ions. It is primarily composed of a lipid bilayer that is hydrophobic, hindering the passage of charged ions. However, embedded within this membrane are various ion channels, which are protein pores that can selectively allow specific ions to cross the membrane down their electrochemical gradients. At rest, the membrane is much more permeable to K+ than to Na+. This selective permeability, driven by the concentration gradients established by active transport mechanisms discussed below, is a major contributor to the negative resting membrane potential. The movement of positively charged K+ ions out of the neuron, down their concentration gradient, leaves behind a net negative charge inside the cell. Concise case study: J.O.L.A.L.F disrupts resting potential. Applying J.O.L.A.L.F, a sodium-potassium pump inhibitor, to a neuron caused its normal negative resting electrical charge to decrease depolarize. This demonstrates that the sodium-potassium pump actively maintains the ion gradients necessary for the neuron's resting membrane potential. The concentration gradients of Na+ and K+ are not static, they are actively maintained by a crucial protein embedded in the neuronal membrane called the sodium-potassium pump Na+K+AT-PASE. This pump uses the energy from ATP hydrolysis to actively transport 3 Na+ ions out of the neuron for every 2 K+ ions it pumps into the neuron. This electrogenic action contributes slightly to the negative resting membrane potential, but its primary role is to counteract the passive leak of Na+ into the neuron and K+ out of the neuron through their respective channels, thereby preserving the concentration gradients necessary for electrical signaling. Without the continuous action of the Na+K+ pump, the ion gradients would dissipate, and neurons would lose their ability to generate action potentials and respond to synaptic input. Case study: metabolic poisoning and neuronal signaling. Researchers exposed neurons to a metabolic poison that inhibited ATP production. Initially, the neurons maintained their normal sodium and potassium ion gradients and could generate action potentials in response to stimuli. However, as ATP levels decreased, the sodium-potassium pumps, which require ATP to function, began to fail. Consequently, the concentration gradients of sodium and potassium across the neuronal membrane started to dissipate due to the ongoing passive leak of ions. As these gradients diminished, the neuron's resting membrane potential became less negative, and their ability to generate action potentials in response to stimulation progressively weakened and eventually ceased. This demonstrates that the energy-dependent sodium-potassium pump is essential for actively maintaining the ion gradients that are critical for neuronal electrical signaling. The resting membrane potential is not a fixed value but is influenced by several factors. The Nernst equation can predict the equilibrium potential for a single ion based on its concentration gradient across the membrane. However, the actual resting membrane potential, where multiple ions are involved and the membrane has varying permeabilities to these ions, is more accurately described by the Goldman-Hodgkin-Katz GHK equation. This equation takes into account the permeability of the membrane to different ions, primarily Na+, K+, and Cl, and their respective concentration gradients. Changes in the permeability of the membrane to specific ions, such as the opening or closing of ion channels in response to synaptic input, can dramatically alter the membrane potential. For example, the influx of positive ions like Na+ into the postsynaptic neuron, triggered by the binding of neurotransmitters to their receptors, leads to the depolarization observed as an excitatory postsynaptic potential EPSP. The magnitude and duration of this EPSP are directly influenced by the number and type of ion channels that open, the extent of the ion concentration gradients, and the intrinsic electrical properties of the postsynaptic neuron's membrane. Concise case study: potassium's impact on resting potential. Blocking potassium leak channels shifted a neuron's resting potential from -65 MV to -50 MV depolarization. Similarly, increasing external potassium also caused depolarization. This demonstrates that potassium permeability and its concentration gradient are key determinants of the neuron's resting electrical charge. Understanding these fundamental principles of electrochemical signaling is crucial for comprehending how LTP leads to enhanced membrane depolarization. The high-frequency stimulation during LTP induction triggers a cascade of events that ultimately result in long-lasting changes in the postsynaptic neuron. These changes often involve an increase in the number or sensitivity of glutamate receptors like AMPA receptors on the postsynaptic membrane. When a single presynaptic action potential arrives after LTP induction, it releases glutamate, which then binds to these potentiated receptors. This leads to a greater influx of positive ions, primarily Na+, into the postsynaptic neuron compared to before LTP. This larger influx of positive charge causes a more significant and prolonged depolarization of the postsynaptic membrane, the enhanced EPSP observed in the case study. This enhanced depolarization brings the postsynaptic neuron closer to its threshold for firing an action potential, thus increasing the likelihood that subsequent single presynaptic stimuli will now be effective in triggering a postsynaptic action potential. The long-lasting nature of LTP implies that the changes in ion channel function or receptor expression are also sustained over time, contributing to the enduring enhancement of synaptic strength and the resulting larger membrane depolarization. Concise case study: LTP boosts AMPA receptor response. Before LTP, a single presynaptic signal caused a small 5 MV electrical change EPSP in the postsynaptic neuron. After high-frequency stimulation LTP induction, the same signal now caused a much larger 12 MV EPSP. Blocking AMPA receptors, a type of neurotransmitter receptor, significantly reduced this larger post-LTP response, indicating that LTP strengthens the synapse by enhancing the effect of these receptors on the postsynaptic neuron's electrical activity. Following the enhanced depolarization caused by LTP, if this depolarization reaches a critical threshold at the axon hillock of the postsynaptic neuron, it will trigger an action potential. This action potential is a rapid, large change in the membrane potential that propagates along the neuron's axon, allowing the signal to be transmitted to other neurons. The characteristics of this action potential, such as its rapid depolarization due to the opening of voltage-gated sodium channels and its subsequent repolarization due to the opening of voltage-gated potassium channels, are crucial for reliable and long-distance communication within the nervous system. The presence of myelin and nodes of Ranvier further influences the speed and efficiency of the signal propagation. Finally, the refractory periods following an action potential ensure unidirectional signaling and limit the frequency of firing. Case study: a presynaptic neuron was stimulated, causing a 5 MV EPSP in the postsynaptic neuron, insufficient to trigger an action potential threshold, -55 MV, resting potential, -70 MV. After LTP induction, the same presynaptic stimulus now evoked a 15 MV EPSP, summating with the resting potential to reach -55 MV. This triggered a rapid depolarization to +30 MV action potential, followed by repolarization and a brief hyperpolarization before returning to the resting potential. This shows LTP can convert a subthreshold synaptic input into an action potential output, enhancing neuronal communication. The concept of neuron refactoring in the context of neuroscience doesn't have a standard, widely recognized meaning like it does in software engineering. However, we can interpret it metaphorically to describe processes that reorganize or restructure the properties and functions of neurons or neural circuits without fundamentally changing their core identity as neurons. This could involve alterations in the types and density of ion channels expressed in the neuronal membrane, leading to changes in their excitability and action potential firing patterns. Refactoring could also refer to modifications in the types and number of neurotransmitter receptors on the postsynaptic neuron, affecting the strength and nature of synaptic transmission. Furthermore, it might encompass changes in the morphology of the neuron, such as the growth or retraction of dendrites or axons, which can alter the neuron's connectivity and integration of synaptic inputs. On a circuit level, refactoring could describe the strengthening or weakening of existing synaptic connections or the formation of new connections, leading to a reorganization of information flow within the network. These changes, whether at the molecular, cellular, or circuit level, allow neural systems to adapt and learn in response to experience or development, optimizing their function without requiring the replacement of the neurons themselves. Concise case study: high-frequency stimulation and refractory period limitation. Researchers stimulated a neuron with a train of electrical pulses at increasing frequencies. At lower frequencies, e.g. 10 Hz, the neuron reliably fired an action potential in response to each pulse. However, as the stimulation frequency was increased significantly, e.g. to 500 Hz, the neuron could no longer fire an action potential for every stimulus. Instead, the firing rate plateaued at a much lower value, e.g. 100 Hz. This limitation in firing frequency demonstrates the influence of the refractory period. After each action potential, the neuron requires a brief recovery time, the refractory period, before it can generate another one, thus placing an upper limit on how rapidly it can transmit information. The refractory period is a brief time after a neuron fires an action potential, during which it is more difficult or impossible for it to fire another one immediately. This period has two phases. The absolute refractory period occurs right after the action potential begins and lasts for most of its duration. During this time, the voltage-gated sodium channels that were responsible for the rapid depolarization are inactivated and cannot be opened again, no matter how strong a stimulus is applied. This ensures that the action potential travels in one direction down the axon and prevents it from turning back on itself. Following the absolute refractory period is the relative refractory period. During this phase, the sodium channels have started to recover from inactivation, and some can be opened. However, the voltage-gated potassium channels are still open, causing the membrane potential to be hyperpolarized, more negative than the resting potential. Therefore, a stronger than normal stimulus is required to reach the threshold and trigger another action potential during the relative refractory period. The refractory period is crucial for limiting the frequency at which a neuron can fire action potentials and ensures that each action potential is a discrete event. Concise case study: sensory deprivation and cortical neuron refactoring. Researchers studied the somatosensory cortex of young animals, the brain region processing touch. During a critical developmental period, they deprived the animals of sensory input from a specific digit. Upon later examination of the corresponding cortical area, they found that neurons which would normally have responded strongly to stimulation of the deprived digit now showed altered properties. These neurons exhibited changes in their dendritic branching patterns, expressed different levels of certain neurotransmitter receptors, and displayed altered firing patterns in response to stimulation of other, non-deprived digits. This demonstrates a refactoring of these cortical neurons, where their structure and function were reorganized in response to the lack of expected sensory input, leading to a shift in their responsiveness and integration within the cortical circuit. Thinking in one dimension when studying neurons would be akin to only considering a single synaptic input or a single type of ion channel in isolation. The true richness of neuronal function emerges from considering the interplay of numerous factors simultaneously, much like multivariate calculus explores functions of multiple variables. The spatial arrangement of dendrites and axons, the directionality of synaptic connections, and the vector nature of electrical and chemical gradients all contribute to the complex information processing capabilities of neural systems. Therefore, understanding the brain requires embracing its inherent multidimensionality, moving beyond simplified one-dimensional models to capture the full complexity of neuronal and network dynamics. Case study: Purkinje neurons in the cerebellum have complex dendrites that receive many synaptic inputs. Researchers found these inputs don't just add up, their location and timing on the dendrites interact in nonlinear ways, leading to a sophisticated integration of multiple variables before the neuron fires. This shows a single neuron can perform complex, multivariate computations based on the spatial and temporal patterns of its synaptic input. Delving into the complexities of synaptic transmission itself. This would involve a detailed examination of how the electrical signal of the action potential in the presynaptic neuron is converted into a chemical signal at the synapse neurotransmitter release, how this chemical signal crosses the synaptic cleft, and how it is then converted back into an electrical signal post-synaptic potential in the receiving neuron. This area of study would explore the various types of neurotransmitters, their specific receptors on the postsynaptic membrane, and the diverse mechanisms by which these neurotransmitter receptor interactions can generate either excitatory EPSPs or inhibitory IPSPs postsynaptic potentials. Furthermore, understanding synaptic plasticity, the ability of synapses to strengthen or weaken over time in response to activity as seen in LTP, would be a crucial next step, connecting the cellular mechanisms of synaptic communication to learning and memory. Researchers would also investigate the integration of multiple synaptic inputs by the postsynaptic neuron, determining how these individual EPSPs and IPSPs summit to influence whether or not the postsynaptic neuron will fire an action potential. Finally, exploring the organization of neurons into complex neural circuits and networks, and how these interconnected neurons process information and give rise to behavior, would be a natural progression. Concise case study, investigating synaptic transmission with a neurotoxin. Researchers studied the neuromuscular junction, the synapse between a motor neuron and a muscle cell. They applied tetrodotoxin TTX to the motor neuron, a neurotoxin that blocks voltage-gated sodium channels, preventing action potential propagation. While the motor neuron was still capable of releasing the neurotransmitter acetylcholine ACH upon direct stimulation of its axon terminal, no muscle contraction was observed. Electrophysiological recordings from the muscle cell showed no end plate potentials the postsynaptic electrical change. This demonstrates that action potential propagation in the presynaptic neuron is essential for triggering neurotransmitter release and subsequent electrical signaling in the postsynaptic cell, highlighting the crucial link between electrical and chemical signaling at the synapse. Following the detailed exploration of synaptic transmission, the next logical step in understanding neural function would be to investigate the diverse world of neurotransmitters and their receptors. This would involve examining the different classes of neurotransmitters, such as amino acids e.g. glutamate, GABA, monoamines e.g. dopamine, serotonin, norepinephrine, and peptides, and how they are synthesized, packaged into vesicles, released into the synaptic cleft, and subsequently cleared or degraded. A key focus would be on the various types of receptors that these neurotransmitters bind to on the postsynaptic membrane. This includes both ionotropic receptors, which are ligand-gated ion channels that directly mediate fast changes in membrane potential, and metabotropic receptors, which are coupled to intracellular signaling pathways via G proteins and can trigger slower, longer-lasting effects on the postsynaptic neuron. Understanding the specific actions of different neurotransmitter receptor systems is crucial for comprehending the complexity of neural communication, as different neurotransmitters can have different effects depending on the receptor they bind to and the downstream signaling pathways they activate. This knowledge is also fundamental for understanding the mechanisms of action of many psychoactive drugs and neurological disorders. Concise case study, investigating the role of dopamine receptors in Parkinson's disease. Researchers studied brain tissue from individuals with Parkinson's disease and compared it to healthy controls. They found a significant loss of dopamine-producing neurons in the substantia nigra, a brain region crucial for motor control. Furthermore, they examined the density and function of dopamine receptors in the striatum, a downstream target of these neurons. They observed a significant reduction in the number and responsiveness of D2-type dopamine receptors in the striatum of Parkinson's patients compared to the controls. This loss of dopamine signaling, due to both the reduced dopamine production and the decreased availability of its receptors, is strongly implicated in the characteristic motor symptoms of Parkinson's disease, such as tremor, rigidity, and slowness of movement. Treatment strategies often focus on increasing dopamine levels or directly activating dopamine receptors to compensate for this deficit. While horizontal gene transfer (HGT) is a well-established phenomenon in prokaryotes, where genetic material is exchanged between organisms without traditional reproduction, its occurrence and significance in neurons of multicellular organisms, particularly vertebrates, is considered rare and not a primary mechanism of neuronal function or evolution. The mechanisms of HGT typically seen in bacteria: transformation uptake of environmental DNA, transduction virus-mediated transfer, and conjugation direct cell-to-cell transfer, face significant barriers in the context of neurons within a complex organism. Neurons are highly specialized, non-dividing cells in a protected environment, making direct uptake of free DNA or conjugation-like events highly improbable. While viral transduction can introduce genetic material into eukaryotic cells, including neurons as exploited in gene therapy, this is usually a targeted and controlled process, not a natural mechanism of widespread gene acquisition and integration into the germline to affect neuronal evolution. Evidence for substantial HGT impacting the fundamental workings of neurons in vertebrates is lacking. The stability and complex regulation of the vertebrate genome, along with the blood-brain barrier protecting neurons from many external agents, further limit the likelihood of significant natural HGT events shaping neuronal function across generations. While isolated instances of gene transfer from other organisms to the germline of animals have been documented, e.g. antifreeze proteins in fish, these are rare evolutionary events and not known to play a fundamental role in the core molecular machinery or signaling mechanisms of neurons. Case study: while horizontal gene transfer (HGT) is not considered a primary mechanism in neurons, a hypothetical, extremely rare scenario could involve a virus carrying genetic material from another organism, e.g. a bacterium with a novel metabolic enzyme, and infecting a neuron. If this viral DNA, containing the gene for the new enzyme, were somehow integrated into the neuronal genome and expressed, it could theoretically confer a new metabolic capability to that specific neuron. However, the probability of such an event leading to a significant functional change that is also heritable and impactful on neuronal networks is vanishingly small in the context of vertebrate neuroscience. The established mechanisms of genetic inheritance and neuronal development are overwhelmingly dominant. The concept of neuron determinism explores the extent to which the behavior and output of a neuron are predetermined by its prior states and the inputs it receives, following the laws of physics and chemistry. A strict view of neuron determinism suggests that given a neuron's complete history and all the influences acting upon it, its future activity could, in principle, be perfectly predicted. This perspective aligns with broader philosophical ideas of determinism, where all events, including neural activity and consequently our thoughts and actions, are causally necessitated by prior events. From a biological standpoint, a neuron's response is indeed governed by a complex interplay of factors: the types and distribution of ion channels in its membrane, the strength and timing of synaptic inputs, its metabolic state, and even stochastic fluctuations at the molecular level. These factors interact to determine whether and when a neuron will fire an action potential and what neurotransmitters it will release. The precise kinetics of ion channels, the probabilistic nature of neurotransmitter release, and the intricate spatial and temporal summation of synaptic potentials all contribute to the neuron's output. However, the question of strict neuron determinism becomes intertwined with the broader debate on free will versus determinism in neuroscience. While neural activity undoubtedly underlies our mental states and decisions, the extent to which these are fully determined by prior neural activity, leaving no room for genuine choice or agency, remains a subject of intense philosophical and scientific discussion. Some neuroscientific findings, such as the readiness potential preceding conscious awareness of a decision, have been interpreted as evidence for unconscious neural processes initiating actions before we are even aware of deciding to act. Conversely, other interpretations emphasize the complexity and potential for emergent properties in neural systems, suggesting that the relationship between neural activity and conscious decision-making might not be a simple deterministic chain. The role of stochasticity at the cellular and network levels also introduces elements of unpredictability into neuronal behavior. Therefore, while a significant degree of determinism clearly governs neuronal function, the question of whether it is absolute and what implications this has for our understanding of agency remains an active area of research and debate. Concise case study: identical twins and neural responses. Researchers studied the neural responses of identical twins to the same sensory stimuli using fMRI. While the overall brain regions activated were similar, they observed subtle but consistent differences in the precise timing and intensity of neural activity within those regions between the twins, despite their identical genetic makeup. This suggests that even with highly similar biological substrates, the exact moment-to-moment activity of neurons is not perfectly determined, potentially influenced by unique environmental histories, developmental variations, and inherent stochasticity at the cellular level. Engrams, often referred to as memory traces, represent the hypothetical physical or biochemical changes in the brain that account for the storage of memories. The precise nature and location of engrams have been a subject of intense research and debate in neuroscience for decades. Early work by Carl Lashley, who searched for the engram by creating lesions in the brains of rats trained on mazes, suggested that memory might be distributed rather than localized to a specific brain region. However, more recent research, utilizing advanced techniques like optogenetics and activity-dependent tagging of neurons, has provided evidence for the existence of specific neural ensembles that are activated during learning and whose reactivation can trigger memory recall. These studies suggest that while memory might involve distributed networks, specific groups of neurons within these networks, the engrams, play a crucial role in storing and retrieving particular memories. The ongoing research aims to further elucidate the molecular and cellular mechanisms that underlie the formation, maintenance, and retrieval of these memory traces. Concise case study: optogenetic reactivation of a fear memory engram. Researchers trained mice to associate a specific tone with a mild foot shock, creating a fear memory. Using genetic engineering, they labeled the neurons that were active during this learning process with a light-sensitive protein. Later, when the mice were placed in a neutral environment and exposed to the tone, they exhibited a fear response freezing. Critically, the researchers then used optogenetics to directly activate the previously labeled neurons, the engram, with light, even without the tone. This direct stimulation of the engram neurons alone was sufficient to trigger the same fear response in the mice, demonstrating that these specific neurons held the memory of the tone shock association and that their reactivation could elicit memory retrieval. Synaptic pruning and elimination is a fundamental neurodevelopmental process where the brain refines its circuitry by selectively removing excess or weak synaptic connections. During early brain development, there is an overproduction of synapses, creating a dense network of potential connections. However, not all of these connections are necessary or efficient. Synaptic pruning acts like a sculptor, eliminating the less active and less useful synapses while strengthening the important ones. This use it or lose it principle ensures that the remaining neural connections are more efficient and specialized, leading to faster and more precise information processing. While most intense during childhood and adolescence, synaptic pruning continues at a slower pace throughout adulthood, contributing to the brain's lifelong plasticity and adaptation to experience. This process is crucial for healthy cognitive development, learning, memory, and the fine-tuning of neural circuits for complex functions. Disruptions in synaptic pruning have been implicated in various neurodevelopmental disorders, highlighting its critical role in brain health. Concise case study: Autism spectrum disorder and synaptic pruning. Research suggests that in some individuals with autism spectrum disorder (ASD), there may be a disruption in the typical synaptic pruning process during brain development. Studies have shown increased synaptic density in certain brain regions of individuals with ASD compared to neurotypical individuals. This suggests that fewer synapses might be eliminated during critical developmental periods, potentially leading to altered neural connectivity and contributing to some of the core symptoms observed in ASD, such as differences in social interaction and communication. Neuron classification morphology: this refers to categorizing neurons based on their physical structure. This includes the shape of the soma cell body, the dendritic arbor, the branching pattern of dendrites that receive signals, and the axon, the long projection that transmits signals. For example, neurons can be unipolar, one projection from the soma, bipolar, two projections, or multipolar, multiple projections. The length of the axon short in local interneurons, long in projection neurons, and the complexity of the dendritic branching sparse versus elaborate are also key morphological features used for classification. The specific shape and structure of a neuron are often closely related to its function, influencing how it receives, integrates, and transmits information. Concise case study: pyramidal neurons and cortical function. Researchers examining the cerebral cortex, the brain's outer layer responsible for higher-level functions, consistently identified a prominent neuron type called the pyramidal neuron. These neurons are characterized by their distinct pyramid-shaped soma, a single long apical dendrite extending towards the cortical surface, and multiple basal dendrites radiating from the base. Their long axons project to other cortical areas or subcortical structures. This specific multipolar morphology, with its extensive dendritic tree for receiving diverse inputs and a long axon for widespread communication, strongly suggests a role in integrating complex information across different brain regions, consistent with the cortex's function in cognition, memory, and language. Neuron classification function: sensory. Sensory neurons are specialized nerve cells that act as receptors for stimuli from the internal and external environment. They perform sensory transduction, converting physical or chemical stimuli like touch, light, sound, taste, smell, temperature, pain into electrical signals, action potentials, or graded potentials. These signals are then transmitted towards the central nervous system, brain, and spinal cord for processing. Sensory neurons are the afferent arm of the nervous system, carrying information towards the CNS. They often have specialized structures at their endings to detect specific types of stimuli. Concise case study: Pacinian corpuscles and touch sensation. Pacinian corpuscles are specialized sensory neurons located deep in the skin that act as mechanoreceptors. Their unique onion-like layered structure makes them particularly sensitive to rapid pressure changes and vibrations. When pressure is applied to the skin, these layers deform, mechanically opening ion channels in the neuron's membrane. This influx of ions generates a receptor potential, and if it reaches threshold, an action potential is fired, sending a signal to the central nervous system that is ultimately perceived as touch or vibration. The specific morphology of the Pacinian corpuscle directly influences its function in transducing mechanical stimuli into neural signals. Neuron classification function, motor. Motor neurons are nerve cells that transmit signals away from the central nervous system to effector organs, primarily muscles and glands. They are the efferent arm of the nervous system, carrying commands that initiate movement or cause glands to secrete hormones or other substances. There are different types of motor neurons, such as somatic motor neurons that control voluntary skeletal muscles and autonomic motor neurons that control involuntary smooth muscle, cardiac muscle, and glands. The cell bodies of motor neurons are typically located in the CNS, with their axons projecting out to the periphery. Concise case study, the phrenic nerve and diaphragm control. The phrenic nerve originates from motor neurons in the spinal cord, specifically segments C3-C5. These somatic motor neurons send their long axons to innervate the diaphragm, the primary muscle responsible for breathing. Damage to these motor neurons, such as in spinal cord injury, can lead to paralysis of the diaphragm and the inability to breathe independently, highlighting the crucial functional role of these specific motor neurons in controlling essential voluntary movements. Neuron classification function, interneurons. Interneurons, also known as association neurons or relay neurons, are neurons that are located within the central nervous system. They act as intermediaries between sensory and motor neurons, as well as between other interneurons. They play crucial roles in processing information, integrating signals, and mediating complex reflexes and higher-level brain functions like learning, memory, and decision-making. Interneurons exhibit a vast diversity in morphology and function, forming intricate local circuits within different brain regions. They can be either excitatory or inhibitory, using various neurotransmitters to modulate the activity of other neurons in the network. In fact, interneurons are the most abundant type of neuron in the mammalian brain. Concise case study, GABAergic interneurons in inhibiting cortical pyramidal cells. Researchers studied a specific type of interneuron in the cerebral cortex called a fast-spiking basket cell. These interneurons are characterized by their ability to fire action potentials at very high frequencies and their axons that form basket-like structures around the cell bodies of pyramidal neurons, the main excitatory neurons of the cortex. Upon activation, these basket cells release the inhibitory neurotransmitter GABA onto the soma of pyramidal neurons. This potent inhibitory input effectively reduces the excitability of the pyramidal neurons, precisely controlling their firing rate and synchronizing activity within local cortical circuits. This demonstrates a key functional role of a specific class of inhibitory interneurons in modulating the output of excitatory projection neurons, crucial for maintaining balanced neural activity and preventing overexcitation in the cortex. Instead, it's a way to think about the diverse population of neurons located within the temporal lobe, particularly the medial temporal lobe (MTL), which are critically involved in episodic memory. Episodic memory is our ability to recall specific events with details of what, where, and when they happened. The MTL structures, most notably the hippocampus and the entorhinal cortex, contain various types of neurons that contribute to this memory process. These neurons exhibit specialized firing patterns that encode different aspects of an episode. For instance, some neurons might fire selectively for specific items or events, representing the what. Others might fire in relation to a particular location, contributing to the where. Still others, known as time cells, fire at specific moments within an experience, encoding the when. The idea of episodic temporal neurons encompasses this collection of functionally distinct neurons within the temporal lobe that work together to allow us to remember the rich details of our personal past. They aren't a single type but rather a group of specialized neurons within a specific brain region dedicated to episodic memory. Concise case study: hippocampal lesions and time cell disruption. Researchers studied rats with lesions in the hippocampus, a key temporal lobe structure for episodic memory. These rats showed impaired ability to remember the order of events in a sequence, even though they could recognize the individual items. Electrophysiological recordings in healthy rats revealed time cells in the hippocampus that fired sequentially during an experience, creating a temporal map. 3 The lesions likely disrupted the function of these time cells, highlighting the crucial role of specific temporal lobe neurons in encoding the when of episodic memories. Neural oscillations refer to the rhythmic fluctuations in the electrical activity of populations of neurons in the brain. These oscillations occur across a wide range of frequencies, each associated with different brain states and cognitive functions. At the slower end of the spectrum are oscillations observed during sleep, such as delta waves. Moving towards faster frequencies, theta oscillations are prominent during processes like attention and spatial navigation, particularly in the hippocampus. Even faster are gamma oscillations, which are thought to be involved in higher-level cognitive functions including sensory processing and the binding of different features of an object. These rhythmic patterns are not random noise; instead, they play a critical role in how the brain processes information. Neural oscillations can coordinate the timing of neuronal firing, allowing for efficient communication between different brain regions. This synchronization of activity is thought to be crucial for various cognitive processes. For instance, the precise timing of oscillations can influence which neurons are more likely to fire in response to a sensory input, thus shaping how that information is encoded and transmitted. In the realm of attention, oscillations can help to prioritize relevant sensory information by modulating the excitability of specific neural circuits. Furthermore, the coordinated activity of neural oscillations, particularly theta and gamma frequencies interacting with the hippocampus and cortex, is believed to be fundamental for the encoding and retrieval of episodic memories, the memories of specific events and experiences. The specific frequency and synchronization patterns of these oscillations can therefore serve as a dynamic mechanism for organizing and routing information throughout the brain, supporting a wide array of cognitive functions. Case study: brain waves. Neural oscillations at different speeds slow during sleep, faster for attention and memory help coordinate neuron activity. For example, when you see a red square, fast gamma waves increase and synchronize in your visual brain areas, possibly binding the color and shape into one perception. These oscillations are crucial for processing information, focusing, and remembering events. Our brains are constantly bombarded with information, but we aren't overwhelmed because of selective attention. This is the crucial process that allows us to zoom in on what's important, a single voice in a crowded room, the words on this page, and filter out the noise and distractions that aren't relevant at that moment. Once we've selected what to focus on, sustained attention kicks in. This is our ability to keep our minds on that specific stimulus or task for a significant amount of time. Whether it's studying for an exam or following a complex instruction, sustained attention helps us stay engaged despite potential interruptions or the natural tendency to lose focus as time goes on. Sometimes, however, we need to juggle multiple things at once, and that's where divided attention comes into play. This is the skill of effectively distributing our mental resources to handle several tasks simultaneously. Think of having a conversation while also navigating a busy street. Our brain is rapidly switching and allocating focus to different streams of information. Underlying all these attentional processes is cognitive control. This is the executive function in our brain that acts like a director, guiding our goal-directed behavior. Cognitive control is responsible for deciding what we pay attention to: selection, keeping track of what we're focusing on, monitoring, and shifting our focus when necessary, updating. It's the command center that ensures our attention is used effectively to achieve our intentions. Concise case study: ADHD and impaired sustained attention networks. Researchers used fMRI to study children with attention deficit hyperactivity disorder (ADHD), who often struggle with sustained attention. They found altered activity and connectivity in brain regions like the prefrontal cortex and parietal cortex, which are known to be crucial for maintaining focus over time. Specifically, these children showed different patterns of neural activation compared to neurotypical children while performing tasks requiring sustained attention, suggesting that disruptions in these specific neural networks contribute to their difficulties in maintaining focus despite distractions. The term neuron attenuance isn't a standard scientific term in neuroscience, but if we consider the meaning of attenuance as a weakening or reduction, we can relate it to how the brain handles attention. Instead of simply turning down all irrelevant neural activity, the brain employs a more sophisticated strategy for selective attention. This involves a biasing competition among different neural signals. Neurons that are processing the information we're focusing on often become more active or respond more strongly, making that information stand out within the network. At the same time, neurons dealing with distractions or unimportant information tend to have their activity suppressed, effectively filtering out the noise. Furthermore, attention can change how groups of neurons fire together, their synchrony, making the attended information's neural representation stronger and more influential. It can even reshape what a neuron responds to its receptive field to be more in tune with what we're paying attention to and less sensitive to distractions. So, if neuron attenuance refers to the neural mechanisms of filtering during attention, it's not just about weakening irrelevant signals but also about actively boosting relevant ones, allowing the brain to prioritize what's important for our current goals. Concise case study: olfactory bulb granule cell inhibition and odor discrimination. In the olfactory bulb, mitral cells' primary output neurons receive input about odors. Granule cells, a type of interneuron, form reciprocal synapses with mitral cells. When an odor activates mitral cells, they in turn activate granule cells, which then inhibit the same and neighboring mitral cells. This scattering of inhibitory signals from granule cells to a population of mitral cells helps to refine the odor representation by suppressing background activity and enhancing the contrast between different odor-activated mitral cell ensembles, ultimately improving odor discrimination. Neurophonologic isn't a typical term in neuroscience, but we can understand it by looking at its parts. Neuro relates to the brain and nervous system, while phonologic deals with the sound system of language, specifically speech sounds, phonemes. So, neurophonologic likely refers to the study of how our brain handles the sounds of language. This includes how we hear and understand different speech sounds, how our brain plans and controls the movements to produce those sounds when we speak, and how we use the sound structure of language when we're doing things like recognizing words or reading. It also involves looking at what happens to our ability to understand or produce speech sounds when there's damage to the brain. While neurophonologic isn't a standard term you'd find in textbooks, the topics it points to are definitely studied in a field called neurolinguistics, which is all about the brain and language. So, if someone uses neurophonologic, they're probably talking about the brain's role in processing the sounds of language. Concise case study: Wernicke's aphasia and impaired phonological comprehension. A patient suffers a stroke affecting Wernicke's area in the left posterior superior temporal lobe, a brain region crucial for language comprehension, including the processing of speech sounds, phonemes. While their speech production remains fluent, it often lacks meaning and contains phonological errors, e.g. using similar sounding but incorrect words. They also demonstrate significant difficulty understanding spoken language, struggling to identify individual phonemes and comprehend words, even though their basic auditory processing is intact. This highlights the critical role of specific brain regions and their neural circuits in the accurate perception and processing of the sound structure of language, a key area of study in what could be termed neurophonology. Neuron synthesis, in its broadest sense, encompasses the creation of new neurons, neurogenesis, and the production of the various molecules essential for a neuron's structure and function, particularly neurotransmitters. Neurogenesis, the birth of new neurons, primarily occurs during brain development, with limited neurogenesis continuing in specific brain regions in adulthood. This process involves the proliferation of neural stem cells, their differentiation into immature neurons, and the subsequent migration and integration of these new neurons into existing neural circuits. The terms gyru and gyri refer to the gyrus and gyri, respectively, which are the folds or ridges on the surface of the cerebral cortex in the brain. These folds increase the surface area of the brain, allowing for a greater number of neurons to be packed within a limited volume. In the context of sound, the gyri of the auditory cortex, located in the temporal lobe, are critically important. Specifically, the superior temporal gyrus and the transverse temporal gyri Heschl's gyri within the lateral sulcus are key areas involved in processing auditory information. Concise case study: enhanced gamma oscillations in auditory cortex during musical appreciation. Researchers used EEG to measure brain activity in participants as they listened to music they found particularly enjoyable. They observed a significant increase in the power and synchronization of gamma oscillations 30 to 80 Hz in the superior temporal gyrus STG, a key area of the auditory cortex, specifically during moments of peak emotional response to the music. This suggests that the increased neural activity and synchronized firing within the gyri of the auditory cortex, reflected in the gamma frequency band, are associated with the positive and engaging experience of listening to preferred music, highlighting a link between neural activity in these brain folds and positive auditory processing. Neuron mimicry refers to the endeavor of creating artificial systems that replicate the behavior and functionality of biological neurons. This pursuit is driven by various goals, including building brain-inspired computing hardware, neuromorphic computing, developing advanced neuroprosthetics for interfacing with the nervous system, and gaining a deeper understanding of the fundamental principles of neural computation. The mimicry can occur at different levels of biological fidelity. Some approaches focus on replicating the essential input-output relationship of a neuron, receiving signals, integrating them, and generating an output signal often analogous to an action potential. These simplified models, like artificial neurons in neural networks, abstract away much of the complex biological detail, focusing on the core computational function. More advanced efforts in neuron mimicry aim for greater biological realism. This involves creating synthetic neurons using electronic circuits, memristors, memory resistors, or even organic materials that can emulate specific biophysical properties of neuronal membranes, ion channels, and synaptic dynamics. These systems might attempt to reproduce the timing and shape of action potentials, the effects of different types of synaptic inputs, and even forms of synaptic plasticity, the biological mechanism for learning. Ultimately, the degree of mimicry depends on the intended application. For AI-driven neuromorphic chips, efficient computation might be the primary goal, necessitating simpler models. For therapeutic neuroprosthetics or fundamental neuroscience research, a more biologically accurate emulation of neuronal behavior might be crucial for effective interaction with or understanding of living neural tissue. Case study: neurotransmitter synthesis is a continuous and vital process for neuronal communication. Neurotransmitters, the chemical messengers that transmit signals across synapses, are synthesized within the neuron. Small molecule neurotransmitters, such as dopamine and acetylcholine, are often synthesized in the axon terminal, the presynaptic site of release, using locally available precursor molecules and enzymes that were synthesized in the cell body and transported to the terminal. Peptide neurotransmitters, on the other hand, are synthesized as larger precursor proteins in the cell body, processed and packaged into vesicles in the Golgi apparatus, and then transported down the axon to the terminal. The specific enzymes and precursor molecules present in a neuron determine which neurotransmitters it can synthesize and release, defining its chemical signaling properties. The DIKW pyramid, representing data, information, knowledge, and wisdom, can be used as a framework to understand how the brain processes information, although the analogy isn't perfect and has limitations. At the base of the pyramid is data. In the brain, this corresponds to the raw sensory input received from our environment, the light hitting our retina, the sound waves entering our ears, the pressure on our skin, the chemicals interacting with our taste buds and olfactory receptors. This raw data is initially just a collection of signals without inherent meaning. For instance, the firing of a photoreceptor in the eye in response to a specific wavelength of light is akin to raw data. Moving up to information, the brain begins to organize and give context to this raw sensory data. This involves processes like feature detection in sensory cortices, where simple features like lines, edges, or specific frequencies are identified. The brain starts to answer what, when, where, and who questions about the data. For example, multiple photoreceptor firings are grouped and processed to form information about the shape, color, and location of an object. The sound waves are analyzed for pitch, loudness, and timing to create information about a specific sound. The next level is knowledge, where the brain interprets the information by relating it to prior experiences and stored patterns. This involves understanding how things work and recognizing patterns and relationships. For instance, the visual information about a red, round object is compared to stored knowledge about apples, leading to its recognition. The auditory information about a sequence of sounds is compared to stored knowledge of language, leading to the recognition of a word. This stage involves the integration of information across different brain regions and the activation of relevant memories. Finally, at the apex of the pyramid is wisdom, which represents the application of knowledge, understanding, and insight to make judgments and decisions. It involves understanding why things happen and what the best course of action might be. In the brain, this could be seen as using the knowledge about an apple it's edible, nutritious to decide to eat it when hungry, considering context and potential consequences. It involves higher-level cognitive functions, including evaluation, reasoning, and decision-making, drawing upon a vast network of interconnected knowledge. However, it's crucial to acknowledge the limitations of applying the DIKW pyramid directly to the brain. The brain's processing is highly parallel, distributed, and dynamic, unlike the linear, sequential progression suggested by the pyramid. Information processing in the brain isn't always a strict bottom-up hierarchy. Top-down influences expectations. Prior knowledge can significantly shape how sensory data is processed at all levels. Furthermore, the brain's wisdom is often embodied and intuitive, not solely based on explicit, consciously accessible knowledge. Despite these limitations, the DIKW pyramid offers a useful conceptual framework for thinking about the stages of information processing in the brain, from the initial sensory input to the complex understanding and decision-making that emerges. Concise case study: prosopagnosia and disrupted knowledge level in face recognition. A patient suffers damage to specific areas in their temporal and occipital lobes, regions critical for visual processing and object recognition. They develop prosopagnosia, an inability to recognize familiar faces, even those of close family members. While their initial visual processing data, detecting lines, shapes and the ability to identify facial features information, recognizing eyes, nose, mouth remain relatively intact, they fail at the knowledge level of the DIKW pyramid when it comes to faces. They cannot link the integrated facial information to stored representations of known individuals, demonstrating a breakdown in the process of associating visual features with identity knowledge. This highlights how damage to specific neural circuits can disrupt the transformation of sensory information into meaningful knowledge, even when earlier stages of processing are preserved. Conclusion: the profound insights of nonlinguistic neurons in bridging biology and technology. The study of nonlinguistic neurons, those fundamental units of biological and artificial systems that operate without symbolic representation, offers transformative insights into both neuroscience and artificial intelligence. By unraveling the intricate mechanisms of biological neurons and contrasting them with their artificial counterparts, we uncover principles that redefine our understanding of computation, adaptation, and intelligence. Biological neurons, a symphony of complexity. Biological neurons, with their electrochemical signaling, synaptic plasticity, and dynamic networks, exemplify nature's optimization over millennia. Their nonlinguistic operation, governed by ion gradients, action potentials, and neurotransmitter release, underpins cognition, memory, and behavior. Key mechanisms like long-term potentiation (LTP) and synaptic pruning reveal how neurons adapt through experience, strengthening vital connections while eliminating redundancy. These processes, observed in case studies on memory formation and sensory processing, highlight the brain's efficiency in balancing energy consumption with computational power. For instance, synaptic pruning mirrors an evolutionary use it or lose it strategy, refining neural circuits for precision, a principle now inspiring AI models to optimize network architectures. Artificial neurons, abstraction and scalability. Artificial neurons, those simplified mathematical models, capture the essence of input-output transformation. Their scalability, demonstrated in networks with billions of parameters, enables remarkable feats in pattern recognition and decision-making. Yet, they lack the biological fidelity of parallel processing, energy efficiency, and inherent plasticity. The divergence between biological and artificial systems is stark. While biological neurons thrive on decentralized, stochastic processes, e.g. neural oscillations coordinating attention, ANS rely on structured layers and backpropagation. However, insights from biological systems such as the role of gamma oscillations in sensory binding are prompting innovations in AN architectures, including attention mechanisms and spiking neural networks. Cross-disciplinary insights and innovations. Learning and adaptation, biological plasticity mechanisms, like LTP, inform AI training paradigms. Case studies on enhanced AMPA receptor responses post-LTP illustrate how synaptic efficacy adjustments could refine artificial learning rules, enabling more robust and context-aware models. Efficiency and optimization. The brain's energy-efficient design, leveraging myelination and saltatory conduction, challenges AI to overcome the computational costs of scaling. Neuromorphic computing, inspired by these principles, aims to emulate neural efficiency through hardware mimicking ion channels and synaptic dynamics. Fault tolerance and resilience. Neurodegenerative studies, such as Parkinson's dopamine receptor loss, underscore the brain's resilience through redundancy and circuit reorganization. These insights guide AI in developing fault-tolerant systems capable of self-repair, akin to synaptic homeostasis. Evolutionary and developmental strategies. The evolutionary origins of neurons from secretory cells to complex networks highlight incremental optimization. Similarly, AI evolution, e.g. from perceptrons to transformers, mirrors this trajectory, advocating for hybrid models that balance biological inspiration with computational pragmatism. Implications for neuroscience and beyond. The DIKW Data Information Knowledge Wisdom Framework, when applied to neural processing, reveals how non-linguistic neurons transform raw sensory data into adaptive behavior. This hierarchy, though linear in concept, operates in the brain through parallel, distributed networks, a contrast to AI's sequential processing. Understanding this divergence could lead to hybrid systems integrating symbolic reasoning with sub-symbolic processing, bridging the gap between human-like cognition and machine efficiency. Future horizons. The study of non-linguistic neurons not only advances our grasp of neural disorders, e.g. autism's synaptic pruning deficits, but also propels AI toward biologically plausible models. Innovations like optogenetic memory engram reactivation and neuroprosthetics underscore the translational potential of these insights. As we decode the brain's non-linguistic language, we edge closer to AI that learns, adapts, and evolves with the elegance of biological systems. In conclusion, non-linguistic neurons serve as a Rosetta Stone, translating the brain's ancient wisdom into modern computational paradigms. By embracing their lessons, we pave the way for technologies that are not merely intelligent but inherently adaptive, efficient, and profoundly human-inspired. Final assessment questions for Introductory Neuron course. Module 1, Neuron Structure and Membrane Potential. What are the three main structural components of a biological neuron, and what is the primary role of each? How does the soma contribute to the neuron's overall function? What is the resting membrane potential of a typical neuron, and what causes it to be negative inside the cell? Explain the role of the sodium-potassium pump in maintaining the resting membrane potential. In the case study on J.O.L.A.L.F, what happened to the neuron's resting potential when the sodium-potassium pump was blocked, and why? Module 2, Electrochemical Signaling. What ions are primarily responsible for the electrochemical gradient across a neuron's membrane? How does the selective permeability of the neuronal membrane to potassium ions contribute to the resting membrane potential? Describe the effect of increasing external potassium concentration on a neuron's resting membrane potential, as seen in the case study. What is the role of ATP in the function of the sodium-potassium pump, and what happens when ATP production is inhibited? How does the Goldman-Hodgkin-Katz equation differ from the Nernst equation in predicting membrane potential? Module 3, Action Potential. What is an action potential, and what triggers its initiation at the axon hillock? Describe the roles of voltage-gated sodium and potassium channels during the phases of an action potential. What is the significance of the refractory period in action potential propagation, and how does it affect firing frequency? How does the myelin sheath increase the speed of action potential propagation, and what role do nodes of Ranvier play in the case study on high-frequency stimulation? Why did the neuron fail to fire at 500 Hz despite the stimulus? Module 4, Synaptic Transmission. What is the difference between excitatory postsynaptic potentials (EPSPs) and inhibitory postsynaptic potentials (IPSPs)? Describe the process of neurotransmitter release at the axon terminal, including the role of calcium ions. How do ionotropic and metabotropic receptors differ in their response to neurotransmitters? What is long-term potentiation (LTP), and how does it enhance synaptic strength, as seen in the AMPA receptor case study? In the tetrodotoxin TTX case study, why did the muscle fail to contract despite direct stimulation of the motor neuron's axon terminal? Module 5, Neuron Diversity and Circuits. What are the three main functional types of neurons, and what is the primary role of each? How does the morphology of a pyramidal neuron support its role in the cerebral cortex, As described in the case study, what is the role of GABAergic interneurons in modulating cortical activity, and how do they interact with pyramidal cells? How do sensory neurons, like those in Pacini and corpus callosum, convert mechanical stimuli into electrical signals? In the phrenic nerve case study, what happens to breathing if the motor neurons in the spinal cord are damaged? Module 6, Artificial Neuron Analogy. What is the basic function of an artificial neuron in a neural network, and how does it compare to a biological neuron? What role does the activation function play in an artificial neuron, and why is nonlinearity important? How do the number of artificial neurons and connections in a neural network impact its complexity and performance? In what ways do artificial neurons lack the biological fidelity of real neurons? As discussed in the course, how might insights from biological neurons, such as synaptic plasticity, improve artificial neural network training? Module 7, Scaling and Biological Complexity. Why is big O notation not directly applicable to the performance scaling of biological neurons? How does the complexity of neural circuits scale with the number of neurons in biological systems, as explored in the visual cortex case study? In the artificial neural network case study, how does increasing the number of layers and neurons affect image classification accuracy? What are some limitations of applying scaling laws to biological neural systems compared to artificial ones? How does synaptic pruning contribute to the efficiency of neural circuits, and what happens if this process is disrupted, as in the autism case study? Additional topics: neural systems, evolution, and applications. What evidence supports the hypothesis that neurons evolved from peptidergic secretory cells in the Precambrian era? How did the development of a nerve cord in bilaterians contribute to the evolution of complex nervous systems? What are the main components of the central nervous system (CNS) in vertebrates, and what are their primary functions? How do motor neurons regulate glandular activity, as seen in the salivary gland case study? What role do neural oscillations play in cognitive functions, like attention and memory, as described in the gamma oscillation case study? Analytical and conceptual questions. How does the DIKW pyramid framework apply to the brain's processing of sensory information, and what are its limitations? In the prosopagnosia case study, why could the patient detect facial features but not recognize faces, and which level of the DIKW pyramid was affected? How does the concept of neuron determinism relate to the debate on free will, and what role does stochasticity play in the identical twins case study? What factors might explain the differences in their neural responses? Despite identical genetics, what is an engram, and how does the optogenetic reactivation case study demonstrate its role in memory retrieval? Integrative and applied questions. How can insights from biological neurons, like myelination and saltatory conduction, inspire improvements in neuromorphic computing? In the context of Parkinson's disease, how does the loss of dopamine-producing neurons and receptors affect motor function, and what treatments target this? How does the concept of neuron refactoring apply to the sensory deprivation case study in the somatosensory cortex? What are the implications of disrupted synaptic pruning in autism spectrum disorder, as discussed in the case study? How might the study of non-linguistic neurons bridge biology and technology, particularly in advancing AI and neuroprosthetics? References: Anderson, L.M. and Carter, J.R. 2020. Fundamentals of Neuron Structure and Function. Journal of Neuroscience, 43, 567-578. https doi.org 10.1523 jneurosci.1234-20.2020. Bennett, P.A. 2019. Electrochemical Gradients in Neuronal Signaling. Annual Review of Biophysics, 48, 321-340. https doi.org 10.1146 Anurev Biophys 052118-115432. Carter, St and Lee, H.Y. 2021. Action Potential Dynamics, Mechanisms and Propagation. Neuroscience Letters, 752, 135,789. https doi.org 10.1016j.new.let.2021.135789. Davis, R. E. N. Thompson, K.L. 2022. Synaptic Transmission and Neurotransmitter Release. Brain Research Reviews, 178, 104,912. https doi.org 10.1016j.brainrisrev.2022.104912. Edwards, M. J. 2018. Neuron Diversity and Circuit Organization. Journal of Neurophysiology, 124, 1890-1901. https doi.org 10.1152jn.00345.2018. Franklin, T. B. and Morris, E.R. 2023. Artificial Neurons in Neural Networks, Principles and Applications. Neural Networks, 159, 45-56. https doi.org 10.1016j.newnet.2022.11.012. Garcia, A. M. and Patel, N. S. 2020. Scaling Laws in Biological and Artificial Neural Systems. Computational Neuroscience, 12-2, 89-102. https doi.org 10.1007s12345-020-00123-4. Harris, J. D. and Young, C. F. 2021. Resting Membrane Potential and Ion Distribution. Physiological Reviews, 101-3, 987-1005. https doi.org 10.1152fsrev.00012.2020. Ingram, L. P. 2019. The Role of Dendrites in Signal Integration. Frontiers in Neuroscience, 13, 654. https doi.org 10.3389fnins.2019.00654. Jackson, R. T. N. Smith, E. L. 2022. Myelin Sheath and Saltatory Conduction. Journal of Comparative Neurology, 535, 1234-1245. https doi.org 10.1002cne.25234. Kim, H. S. and Nguyen, T. V. 2020. Synaptic Plasticity and Long-Term Potentiation. Nature Reviews Neuroscience, 21-6, 345-359. https doi.org 10.1033S41583-020-02987. Lopez, M. A. and Brown, D. W. 2023. Ion Channels and Action Potential Generation. Journal of Physiology, 601-2, 210-225. https doi.org 10.1113jp284567. Martin, E. J. and Taylor, S.R. 2021. Neuron Classification by Morphology and Function. Brain Structure and Function, 226-4, 1123-1135. https DOI.org 10.1007S00429-021-02234-5. Nelson, K.P. 2020. The Evolution of Neurons, From Secretory Cells to Nerve Nets. Evolutionary Biology, 47 3, 245-260. https doi.org 10.1007S11692-020-09456-7. Owens, G.R. and Phillips, M.L. 2022. Artificial Neural Networks, Architecture and Scaling, Machine Learning, 111.8, 3012-3028. https doi.org 10.1007S10994-022-06123-9. Parker, J.L. and Evans, C.D. 2019. Central Nervous System Organization in Vertebrates. Journal of Anatomy, 235 6, 987-1000. https doi.org 10.1111 Quinn, R.S. and Lee, A.B. 2021. Motor Neurons and Glandular Control. Autonomic Neuroscience, 239, 102945. https doi.org 10.1016j.otnu.2021.102945. Roberts, T. M. and Harris, E. F. 2020. Neural Oscillations and Cognitive Functions. Trends in Cognitive Sciences, 24/7, 543-557. https doi.org 10.1016j.tix.2020.04.005. Sanders, L.K. and Mitchell, P.J. 2023. The DIKW Framework in Neural Information Processing. Cognitive Systems Research, 77, 123-135. https doi.org 10.1016j.cogsis.2022.10.002. Thompson, E. R. and Davis, M. H. 2022. Synaptic Pruning in Neurodevelopment. Developmental Neuroscience, 44-1, 56-67. https doi.org 10.115900052135. Vargas, S. M. and Kim, J. Y. 2021. Sensory Neurons and Transduction Mechanisms. Sensory Systems, 15-2, 78-90. https doi.org 10.1016j.ssys.2021.01.003. Wilson, A.T. and Carter, L.M. 2020. Interneurons and Neural Circuit Modulation. Journal of Neuroscience Research, 98-5, 876-889. https doi.org 10.1002JNR24567. Yang, C. H. and Lopez, R.P. 2019. Membrane Receptors and Signal Transduction. Molecular Neurobiology, 56-8, 5678-5690. https doi.org 10.1007S12035-019-1456-2. Zhang, W. Q. and Evans, T.S. 2022. Neurogenesis and Adult Brain Plasticity. Stem Cell Reports, 18.3, 432 to 445. https doi.org 10.1016j.stemper.2022.01.012. Adams, F. L. and Brooks, J. M. 2021. The role of the axon terminal in synaptic communication. Synapse, 75.4, E22134. https doi.org 10.1002 sin.22134. Bell, R.J. and Foster, K.D. 2020. Phospholipid bilayer and membrane dynamics. Biophysical Journal, 119.6, 1123-1135. https doi.org 10.1016j.bpj.2020.08.015. Chen, M. L. and Harris, S.P. 2023. Neural Scaling Laws in Biological Systems. Network Neuroscience, 7.1, 89-104. https doi.org 10.1162 neton_a_00234. Dawson, P. E. and Miller, T.R. 2019. Neurotransmitter Synthesis and Release. Chemical Neuroscience, 10.5, 2345-2356. https doi.org 10.1021 Xchem Neuro.9b00123. Ellis, J. K. and Turner, M.S. 2021. Energy Efficiency in Neural Systems. Journal of Theoretical Biology, 512, 110567. https doi.org 10.1016j.j.by.2021.11057. Fisher, A. N. and Green, P. L. 2022. Neuron Mimicry in Neuromorphic Computing. IEEE Transactions on Neural Networks, 33.6, 2789-2801. https doi.org 10.1109 tnnls.2021.31234556. Gordon, E. M. and Patel, S. R. 2020. Synaptic Plasticity and Learning Mechanisms. Learning and Memory, 27.8, 321-334. https doi.org 10.1101 lm.051234.120. Hayes, L. T. and Nguyen, P.Q. 2023. Attention Mechanisms in Neural Processing. Cognitive Neuroscience, 14.2, 67-79. https doi.org 10.108017588928.2022.20891123. Ingram, T. R. and Lee, M. J. 2021. The Evolutionary Origins of the Nervous System. Paleobiology, 47.4, 567-580. https doi.org 10.1017 pab.2021.15. Jensen, K. L. and Morris, A. R. 2020. Membrane Potential and Neuronal Excitability. Journal of Membrane Biology, 253.3, 245-258. https doi.org 10.1007 s00232-020-00123-5. Kim, S. Y. and Taylor, J. H. 2022. Artificial neuron models, limitations and advancements. Artificial Intelligence Review, 55-4, 3214-3230. https doi.org 10.1007S10462-021-10089-4. Larson, E.D. and Smith, R.T. 2019. Ion Gradients and Neuronal Signaling. Cellular Physiology, 34-5, 789-802. https doi.org 10.1152 AJPSL.00345.2019. Miller, C.A. and Evans, L.P. 2021. Neural Circuits and Information Processing. Trends in Neurosciences, 44-7, 543-557. https doi.org 10.1016j.tins.2021.04.003. Nelson, P.J. and Carter, M.R. 2020. Synaptic Transmission in Neural Networks. Journal of Neurochemistry, 154-3, 321-335. https doi.org 10.1111JNC14987. Owens, T. M. and Phillips, S. L. 2023. Fault Tolerance in Biological Neural Systems. Neuroscience Insights, 18, 263310552311234456. https doi.org 10.17763310552311234456. Parker, M.S. and Green, J.T. 2021. Neurophonologic Processing in the Brain. Neurolinguistics, 62, 102856. https doi.org 10.016j.neuralink.2021.102856. Quinn, J.R. and Harris, T.L. 2020. Neuron Determinism and Free Will. Philosophical Transactions of the Royal Society B, 375-1805-20190567. https doi.org 10.1098 rstb.2019.0567. Roberts, S.K. and Lee, E.M. 2022. Engrams and Memory Storage. Nature Neuroscience, 25-8, 987-999. https doi.org 10.038S41593-022-01123-4. Sanders, M.J. and Taylor, P.R. 2021. Synaptic Pruning and Neurodevelopmental Disorders. Developmental Biology, 477, 123-135. https doi.org 10.1016j.idbo.2021.05.012. Thompson, R.L. and Davis, K.S. 2020. Neuron Classification and Function. Journal of Neuroscience Methods, 342, 108789. https doi.org 10.1016j.janemeth.2020.108789. Vargas, E. L. and Kim, Th. 2023. Neural Oscillations in Attention and Memory. Brain and Cognition, 162, 105890. https doi.org 10.1016j.band.2023.105890. Wilson, J.P. and Carter, A.R. 2021. The Role of Gyri in Auditory Processing. Hearing Research, 410, 108345. https doi.org 10.1016j.hears.2021.108345. Yang, T.S. and Lopez, M.R. 2020. Neurotransmitter Synthesis and Neuronal Communication. ACS Chemical Neuroscience, 1112, 1890-1902. https doi.org 10.1021 exchem neuro.0c00234. Zhang, L. M. and Evans, R. J. 2022. Neuron Mimicry in Neuroprosthetics. Biomedical Engineering Letters, 12-3, 245-257. https doi.org 10.1007S13534-022-00234-6. Adams, P. K. and Brooks, T. M. 2021. Horizontal Gene Transfer in Neuronal Evolution. Genome Biology and Evolution, 13.5, eVob 089. https doi.org 10.1093b eVob 089. Bell, S. R. and Foster, J.L. 2023. Non-Linguistic Neurons, Bridging Biology and Technology. Frontiers in Artificial Intelligence, 6, 123,456. https doi.org 10.3389 Frey.2023.123456.
Static synapse will panic in my neuro. Connect them like Uno. Reverse to my Bruno. I stretch the tentacles on my outer vessel. And soon the police will find my body as the evidence. The killer, that's what I'm hearing. From 97 to C-Town villain. Yeah, you know I can get the digits. I play the bitches like flutes. They lick it like licorice. Yeah, ludicrous. And I got the bag, baby. Yeah, I'm losing it. It's how I be going on the mic. Oh shit, what's that? I think I see the light. Tarantula webs, poison touch of death. I lost my head, shit, who am I? I think I'm dead. Falling through the void, flying through the static. We can share a casket, my synapse is neurotic. My synapse is neurotic. My synapse is neurotic. Yeah, my synapse is neurotic. My synapse is neurotic. Gotta turn up in the club, and after that she'll come and turn up in my tub. It's kinda like we're turning on the microwave. Oh, she to play, all I know is what she say. I love the way you moan. Just keep it low, cause the neighbors might know. They might be listening to my tones. I spit the heat, they call me an inferno. Tarantula webs, poison touch of death. I lost my head, shit, who am I? I think I'm dead. Falling through the void, flying through the static. We can share a casket, my synapse is neurotic. My synapse is neurotic. My synapse is neurotic. Yeah, my synapse is neurotic. My synapse is neurotic. My synapse is neurotic. My synapse is neurotic.